as I was saying, plant breeding is facing really exciting times because we are entering a new era in which selection decisions more and more often depend on predictions. And well, as you may know, genetic recombination occurs at random, so we have very little control about the specific places where this will happen. And therefore, the more genotypes we evaluate in the progeny, the larger the probabilities that at least one of them will have the genetic combinations that will confer adaptation to the target environments. Well, I borrowed these slides from a paper from Mark, and I like it really much because it illustrates the new configuration of modern plant breeding programs that basically rely on prediction, because in early breeding stages, a large number of genotypes are evaluated, so the scale of plant breeding programs has increased enormously in the recent years, and a few of them is characterized phenotypically. And this phenotypic information is used to train statistical models or crop growth models to predict the performance of those genotypes that were not evaluated. Of course, when we advance in the breeding stages, the amount of phenotypic information that is available increases, and the final selection decisions are always made based on information from multi-environment trials. In agricultural system, we can have different sources of variation that need to be accounted for. So one of the first one is the spatial variation, that could be the within trial variation. Think about the trends within the field, for example. We also have spatial variation that is to the locations, so the geographical regions in our target population of environments. The temporal variation can be allocated to year variations for one specific location, but also the within season variation. So the timing of the stress is really important and determines the environmental quality and which genotypes would be adapted to that environmental type. Then, of course, we have the genotype that they have different sensitivities to the environmental conditions, and that leads to this genotype by environment interaction. And some uncertainty about the traits that are relevant to confer adaptation at a specific environment. So here we see that trait correlations change as a function of the environment and over time. When we come to modern plant breeding programs, more and more often we have high throughput phenotyping, and that introduces a new source of error, because usually these technologies rely on proxies of the traits. Think about NDVI or image analysis that are not a direct assessment of traits of like biomass or spike count, but are an approximation. And therefore, there is a new source of error that is related to the measurement error. There are many ways and statistical models to capture these sources of variations. I will not enter into detail, but basically this is the classical way in decomposing into genotype, the variation due to genotype by location, year, and management conditions. But the availability of new genetic and phenotypic variable and environmental variables allows us to decompose G by E into something that looks a little bit complicated like this, but it's nothing else that we want to explain part of the G by E into some covariables that are a function of molecular markers and something that is dependent on environmental conditions. Think about temperature, an index that characterizes water stress, for example. And then there we have some, and this could be the same, but for variables that are related to agronomic management, for example. And of course, there is one part that we cannot explain by the covariables, and that can be used, for example, in statistical models that focus on the variance-covariance structure. So this is always an implicit representation of G by E. How can we do, how can we characterize the environments? Well, here, to understand a little bit more the structure of this part, well, there are many old models like the AMI model, for example, DGGE model, that are statistical procedures to identify groups of environments and to identify which genotypes would perform better under which environmental conditions. And nowadays also there could be the alternative of using crop growth models as a way to classify the environmental conditions at a given trial. And these crop growth model covariables could be introduced in this part of the mixed model. So that was about the commonly used statistical models. We also need to say a little bit about crop growth models. So in here, well, I feel I'm a bit preaching to the Pope because many of these models were developed here in Australia. So here we were working together and following up on the paper by Karine Chenou, 2013, where she used the APSIM model to characterize the drought deficit, the water deficit pattern across the Australian wheat belt. And what can we learn from this type of approaches? Because it allows to characterize the probability of a certain stress to occur at a particular location. So that defines the type of environments that we need to breed for and the traits that will be adapted for that environmental condition. So what do we propose now? Actually, we are saying statistical models are very useful because they are they have a natural way of accommodating uncertainty. If you think about Bayesian model or mixed models, so through the variance-covariance structure. So that's the advantage of mixed models or statistical models. But for them, it's a little bit challenging to model all the sources of variation simultaneously, plus the variation over time. So that doesn't come in a sort of obvious way on how to do it. On the other hand, we have crop growth models that for this type of model is very natural to accommodate temporal variation because this is the way they work at a daily time step. So they are built for that. But in the way they were built, it's not so easy, or it will require a lot of phenotypic data to parameterize them for a large set of genotypes. So that's kind of a constraint that is a more practical constraint. And the last thing that they have the challenge is that they are fully deterministic. So every time we run APSIM with the same weather and with the same set of parameters, we would obtain the same yield. So we want something in between, and we actually want to borrow the positive things from both types of models. And we want to make a combination between statistical, genetic, and crop growth models to characterize the response of many genotypes over time and multiple traits and multiple environments. So what did we do? Well, basically, we had a sample of the target population of genotypes, so the genotypes that are relevant for the wheat genotypes that are relevant for the Australian wheat belt. So this was 200 genotypes for which we had real SNP data. On the other hand, we had a sample of the target population of environment. So based on the work with Karine and Scott and Banju, we selected a number of trials. So it was 4 locations and 31 years of real meteorological data for important locations for wheat in Australia. And what we generated was a population that was segregating for 12 APSIM parameters that regulate the response to the environmental conditions. So from this part onwards, everything is simulated. So how did we simulate these parameters from the real SNP data? Well, we went back to some other trials for wheat in Australia, and we ran GWAS, and we estimated the additive effects. And we say, well, the distribution of the additive effects for wheat in Australia looks like this. So it's a gamma distribution, so many SNPs with small effect and a few with larger effect. And we assume that would be a realistic distribution for our simulated traits. But because we had 12 traits and we wanted to make some of them correlated, we introduced some restriction. And we did that using something that is called copulas, that basically for some traits, so for some pairs of traits, the additive effects regulating the parameters would be correlated. And in that way, the final, the APSIM parameters would show some correlation as well. Then we attach those effects to this real SNP profile, and in that way, we generated the 12 genotype-dependent APSIM parameters. So we go back to our scheme. We have for each genotype a different value for each of the 12 APSIM parameters. Then we introduced these parameters in the model with the meteorological data, and we generated phenotypic information for each genotype, each day, and each environment. But APSIM is fully deterministic, so if we want to evaluate statistical models and their ability to capture G by E, we need to introduce some error, otherwise it's not a realistic setup. So for that reason, we introduced two sources of error. One is the plot error, because of the differences between replicates, for example, and a measurement error, because in that way we can evaluate different phenotyping strategies. So what did we do with this data? So it's large amount of data, remember, for each genotype every day and each of the 124 environments. So the first thing we wanted to look at was how well did we generate G by E? Was it realistic enough so that it would help us evaluating models that could be helpful for real data? So the first thing, well, basically we repeated a bit the approach that has been done here for sorghum and for wheat. And this is the water supply-demand ratio over time. And each line represents a different year in a particular location. So if the water supply-demand ratio is reduced, it means the plants are under stress. So the temporal, the time course of these water supply-demand ratios was used to cluster environments and into four classes that coincide with what was proposed in the previous papers. And we could see that basically locations have a different probability of encountering a certain environment type. For example, in emerald, very often we observe intense stress that is starting early in the growing season, whereas for Yanko, for example, the drought is a lot less frequent. The second question is, well, we have our environment classification and we want to know whether the G by E patterns that are observed in a simulated yield are realistic and whether we can relate them to the water stress conditions. So here we have an AMI plot. So it's basically a singular value decompositions on the interaction. So each of the dots represents a different genotype, and each vector represents one environment. And the color of the vector indicates the environment type. So we see that, for example, all the environments where no stress was observed were highly correlated, and the same for the other groups. So apparently, using the water dynamics to classify environment does a really good job in capturing most of the G by E. Why is this useful? Because we wanted to develop different statistical models to predict individual trials. And we can do that in different ways. So the first way that we use, say, okay, we have 4 environment types, and we will assume that each trial is just a sample from that environmental condition. So we are not interested anymore in predicting each individual trial, but the mean of the environment type. And this was this model that we use here. Then we had a second model that is more, has more biology inside, if you want, because we said, well, if we have traits like radiation use efficiency, the sensitivity to photoperiod, or vernalization, we can as well use them as genotypic covariables to help us understanding G by E, because these will modulate the response to the environmental conditions. So we do a two-way variable selection, on one hand checking which APSIM parameters are relevant, and then checking which environmental covariables are also explaining the differences in genotypic sensitivity to the environment. So on the right-hand side, I'm only showing for 2 locations, but the results were similar for both. We see that actually when we predict the mean of the environment type, we do quite well. So this means on one hand that the environment classification did a good job, and that the groups are internally homogeneous. So basically, we can replace one late drought trial by another late drought trial. They are more or less similar. The second best model was in general, so these are the different years, the factorial regression. So apparently, in this case, the environmental covariables and the APSIM parameters did a good job in explaining G by E. But in practice, so these are simulated, but in practice, you could as well use things measured in your trials. Like I think Banju was working a lot on estimating these two from managed stress trials, well, stress, in which you had long photoperiod, short photoperiod, vernalized and not vernalized. So this could be coming from real trials. And these two didn't do a very good job, but this is basically just constructing a similarity matrix with all the environmental covariables and all the SNPs, so not really a lot of variable selection was going on here. And well, they do not really a good job. So, but so here this was just an application on how simulated data could be insightful and developing some strategy for statistical modeling of G by E. A third application that we implemented for this data was to use it to understand or to develop network methodologies. Because if you have real phenotypic data and you measure many traits in your trial, it's very hard to understand how causality is propagated between your genotype, the different basic mechanisms until final yield. So there are many steps in between, and it's very hard to understand what is causing what, because many times things are correlated to each other. But if we were to intervene, so if we need to apply a selection pressure, or if we would apply something like CRISPR-Cas and fixate flowering time at a certain value, for example, all these are interventions that can be done through breeding, then we would like to know what happens to the other traits, conditional on that value for yield, for flowering time, for example. So in this case, it was easy because we knew the causal flow. We know that the SNP caused the APSIM parameters, and we know that the APSIM parameters cause biomass, and that biomass has an impact on final yield. So this is represented here by the gray dots. So everything that is genotype dependent but constant over time and across environments is represented in gray. And the green dots represent things that are a result of the APSIM parameters and the environment. So what do we see? Basically, we have two networks for drought and non-drought. And we see that, for example, this parameter that is related to the lower limit for water uptake, so how effective are the plants in taking out water from the soil, has a very large influence on biomass and the drought. But this influence is not relevant when drought is not present. So in that way, the topology of the network is also a way of classifying environment, because it tells us about which mechanisms are adaptive and could be selected for in a breeding program. So far I have been talking about traits that are static. So we look at them, biomass at the end of the growing season, etc. But we don't need to forget, we cannot forget that traits are actually dynamic. And now characterizing dynamic traits is a lot more accessible thanks to high throughput phenotyping. So this is not impossible anymore. So what can we learn from trait dynamics? Here I'm showing you, for example, the correlations between the secondary traits and yield. So this is the correlation between biomass and yield for the three different environment types. And the first thing that we see that is very evident, that trait correlations are environment dependent and also time dependent. For example, in this case, we have a positive correlation, but when drought starts, this correlation changes over time. And it also allows us to identify which are the mechanisms that are responsible for that changes or in the importance of biomass for grain yield. So this is just to have a look and to propose which traits could be important for a selection strategy, for example. Another thing that we could do, for example, if we are interested in understanding the underlying QTLs, we could have an explicit formulation for the QTLs over time. So here, these are the additive effects. So these are the QTL for biomass at each single day. And we see that these QTLs for biomass also coincide with other traits that are more basic mechanisms. So for example, a high biomass is related to also a high radiation use efficiency, which of course would make a lot of sense. But the advantage here is that we can identify better which SNPs are responsible for that causal flow between the basic mechanisms and traits that integrate more processes during the growing season. So that's something that we can learn from this. And the last application that I will show, and this is more in the context of high throughput phenotyping. So nowadays, I think everyone would agree here, or almost everyone, I hope, that it's really a lot of, these technologies are really a lot of fun. I hope Anju still agrees. The drones and this kind of, yeah, they are. Everyone gets very excited. I love them as well. But the question is what to do, or when are they useful for a plant breeding program? So we think, if we want to be very pragmatic and say, okay, we will only be phenotyping additional traits if we can obtain better predictions for our target trait, grain yield, then we need to consider this relationship, that this is basically the genetic correlation between the secondary trait and the target trait, so the genetic correlation between biomass and yield, for example, and the heritability of biomass. So if we cannot measure biomass in a very precise way, we would be introducing noise, and then it might not be advantageous to include it in a multi-trait prediction model. So if we go a few slides back, you might remember this is the correlation between biomass and yield. We saw that this is environment dependent, so it's positive when there is no drought. And there is a lot of changes in the intensity of the correlation when there is drought. So it starts negative, and then it ends up being positive. So what can we learn from this? Is it really useful to phenotype for biomass under drought or not? It's hard to know a priori, and this is why we think that these simulations could be useful. So that's related to this part. And then in relation to the second part of the heritability of this relationship, when we have phenotyping devices, it's quite common that they, to see that they have a low heritability, so a large measurement error. So we want to know how can we improve on that with modeling. So basically, we propose that it's very important that to model time points over, to model observations over time. And then you can extract parameters that describe the whole dynamics, things like the asymptote, the maximum rate, or the inflection point, and use those as correlated traits instead of the raw observations for biomass, for example. Why? Because in general, these estimates from the curve dynamics would have a far larger heritability than the single time points. And this is what we show here. So we wanted to know how precise and how often do we need to measure additional traits to improve yield prediction. So when is biomass instrumental for yield prediction? So we tested with simulations different schedules, one measuring in 25, 15, or 5 days, and assuming a range of heritabilities for individual time points. So assuming more deviations here or smaller deviations here. And the results are shown on the right-hand side. So here we have the heritability of single time points, ranging from 0.1 to 0.9. We see, of course, if each time point is measured more precisely, the curve parameters are more precise as well. That's quite logical. But maybe what is less logical is actually there is a kind of compensation mechanism between the frequency and the single time point heritability. This tells us if our device, our NDVI, our phenotyping technique has a limitation that we cannot improve on that heritability, we might as well measure more often and model traits over time, and that would allow us to reach an improved heritability for the curve parameters. So we got rid of part of this measurement error and plot error. And the question is, does it help to improve yield prediction? Because this is our final goal. So here I'm showing 3 figures, one per environment type. And we see that the heritability of the horizontal line shows prediction accuracy for single trait, so predicting yield from yield. And the dots show predicting yield from yield and biomass parameters, so the parameters of biomass dynamics. And we see that in most of the time, it's convenient to model both traits simultaneously, so there is an improved prediction accuracy. Only in few cases, this doesn't work, and basically this tells us we were measuring too few time points, so we need to measure more often. Or in this case, well, probably a heritability of 0.1 for biomass is just way too low. Even if we do all the modeling to get rid of that error, we won't manage. So that's also a logical result. And again, the improvement in accuracy is larger when biomass and yield are correlated. Remember, when there is drought, there was a negative correlation at the beginning and positive at the end. For late drought, the correlation was quite low, and for no drought, the correlation was always positive. So this is the most, the case in which we see the largest advantage. Okay, so that was the last application that I wanted to show for this type of simulated data. And so I hope that with these applications, I was able to explain why we think that the combination of statistical models and crop growth models offers good opportunities to developing modeling strategies and phenotyping strategies, and to predict adaptation across environments. And well, here we cover different questions. So the characterization of the target population of environments and the occurrences of the stress types. Then the trait correlations, which traits are helping to confer adaptation at each environmental conditions. And then, well, this is more in relation to the frequency and heritability that we would need for a phenotyping strategy. So I would like to leave it there, and I would like to thank well, my team members and the UQ people. I also thank you. You are co-authors of both papers, so I thank for your contributions, because I think that shows that there is the synergy between both types of background statistics and crop growth modeling. So now, if you have questions.